Thanks for that kind introduction. I'm very excited to present this work that we've been doing on large-scale rare variant association testing. And I'd like to convince you in this talk that exome and genome sequencing to discover rare variants provides new opportunities to discover causal mechanisms for human diseases. So decades of GWAS, as many of you I'm sure know, have successfully identified thousands of robust loci for common diseases, but they require additional work to get at the causal factor for those diseases. In parallel, Mendelian disease studies have discovered high impact rare variants for rare diseases. And these are really impactful, but these are only finding these only being identified for these rare diseases. Not much work has been done for rare variants for common diseases. It's still somewhat of an open question how much of the disease heritability of common diseases can be explained by rare variants, but we know that we can gain true biological insight from rare variants. But we have in common diseases, but we haven't really explored this systematically. For instance, here in some disorders such as schizophrenia, shown here on the right, early insights from rare variants have implicated dozens of loci, and we're beginning to fill out the spectrum of rare and intermediate frequency variants that contribute to these diseases. So on the x-axis of this plot is the minor allele frequency of a variant in the general population. Then on the y-axis is the odds ratio, or the effect size of the variant that we discover in this association test. So on the kind of right tail here, you can see these common variants that have been discovered by GWAS for schizophrenia. Some factors that you may know and are pretty famous, like complement 4, and SLC39A8 is also a pretty well-known one. And then in parallel, these kind of more rare variant studies are beginning to come online, mostly looking at rare copy number variants. So you can see a lot of these duplications and deletions, although some rare protein truncating variants as well are being implicated. And while these have lower frequencies, they tend to have higher effect sizes in order for them to be discovered in the first place, and then on top of that to be kind of robustly associated. It stands to reason that there's a curve here between these kind of variants, that there's probably some intermediate frequency variants, but they are harder to discover because you need much larger sample sizes because their effect sizes are quite a bit lower. And then on top of that, there are probably plenty of variants also in here, but these are extremely hard to discover because they're both rare and have a relatively small effect size. So we now have even more opportunities to explore these types of questions in large biobanks. This was an example of schizophrenia, but we'd love to do this for a lot of phenotypes as well. And by the way, I should say, feel free to interrupt if you have any questions. I'm more than happy to take them, questions as I'm going, or later on is fine as well. So the UK Biobank is one such example that is widely used in our community due to its highly open access model. When I looked a few months ago at this point, it had been used in over 2,000 publications and showed no sign of slowing down and only really accelerating over the years. The huge advantage of this resource is that there's matched genetic and phenotypic data with a wide variety of phenotypes, including disease outcomes from health records, measured traits, and questionnaires. And there have been plenty of efforts that have explored the association between common variants in many of these traits, including efforts in the local community here at the Broad in the Neal Lab. And more recently, we've embarked on an effort to use all of the available data from individuals of multiple diverse ancestries in the Pan-UKB project in collaboration with Alicia Martin and others. However, today I'll talk about a new opportunity we had to explore the rare variants in this data set through a partnership with several pharmaceutical companies. I should point out a lot of the phenotypic work that was done here was run by Duncan Palmer using a tool called Pheasant, which kind of cleans up a lot of the phenotypic data. While a lot of it is very structured, there's still a lot of kind of QC that needs to be done with this phenotype data. And so here I'm just showing an example of the pipeline of Pheasant and how it kind of considers different types of variables and when they, when they behave in certain ways, what kind of what kind of QC we perform on them. This is, by the way, all of this data, as mentioned, this was all published in medRxiv, or there's a preprint on medRxiv last year where this is all available. All this data is available in the supplement. I'm happy to share that if needed. But yeah, so as I mentioned, this is a partnership with 3 pharmaceutical companies, AbbVie, Biogen, and Pfizer, who funded the processing, quality control, and analysis of the exome data. By the end of the project, the data sciences platform here at the Broad had processed about 450,000 samples which were QC'd by Grace Tiao and her team. And just to give you an idea, this data set was about 3 times larger than the previous largest exome call set, Nomad V2. And this team actually pulled together this data and returned about 20 terabytes of data to our pharma partners and back to the UK Biobank. So really, this was a huge engineering effort to do the processing and quality control of all this data and make sure that it is as high quality as possible to go into the association tests. Of course, their work is not done yet, and we're looking forward to a new release of Nomad. These folks are also on the Nomad team that will coming sometime this year that will integrate these samples, both the Nomad samples and the UK Biobank samples and more. That will be the largest version of Nomad that we have so far, coming sometime this year. Just to give some examples, there's plenty of QC steps that we're go on along the way. I'm not going to show all of them, but for instance, here you can see where when we actually look at these samples, you can we have to a priori from the genetic data assign sex chromosomes to the individuals. This is mostly as a QC check to make sure there are no sample swaps, but also just to kind of be certain about the quality of our data as well. This is a good estimate of contamination samples as well. So you can see there's a nice separation between, in this case, the females and the males, with some sex aneuploidies as well, as expected in these extremely large data sets. When you get up to about 500,000 individuals, you do expect to see quite a bit of individuals who are not XX or XY. So in total, I mentioned 450,000 samples. Unfortunately, I'm not yet able to present that today. We're still pulling together the final data set, but the data set that I'll present in this talk and that's on medRxiv right now is the second to last release of the data at about 300,000 individuals. We do plan to release the full 450,000 data set as well sometime this year, probably in the next few months, but we're still kind of getting permissions and final everything in place for that. So at the end of the day, we had about 281,000 individuals that were high quality samples and in this non-Finnish European cluster so that we can have good high quality data for association testing. So as part of this effort, we had to redesign how we might do ultra large scale association testing. As this pipeline takes about 4,000 CPU hours or about 6 CPU months per phenotype. So that wouldn't be such a big deal for one or a few phenotypes, but for 4,000 of them, this required a whole lot of computing power. Thanks to the Hale team, who built an incredible system for highly parallel analysis using arbitrary programs known as Hale Batch, we were able to run this pipeline using advanced linear mixed models implemented in Sage Gene. This system, which can reliably run tens of thousands of CPUs and even got to about 100,000 simultaneous CPUs at its peak, allowed us to finish this analysis in less than 2 weeks, which is pretty incredible for something that would have taken, that was in the millions of CPU hours, in the tens of millions of CPU hours. If folks have a Broad affiliation, this system is now available to all Brodies. So if anyone's looking for a framework to do large scale analysis at the Broad, I encourage you to check out this website, hale.is, and see if Batch can help you. I think there is a plan to make this even more broadly accessible in the future, but it is still a work in progress. We performed millions of single variant tests per phenotype, but with rare variants, one thing we could do to increase power here was to group variants with the same annotation type in the same genes into a group test. So for instance, all LOF variants in one predicted loss of function variants in one gene, all missense variants and all synonymous variants. And so for each of these, we actually ran several different types of tests as well, including these burden tests, which are the more classical type tests that you would think of, as well as hybrid variance tests in the form of SCAT and SCAT-O for each phenotype. We used a set of simulated data sets to estimate an empirical p-value threshold for what we believe is experiment-wise significance. Because rare variants aren't exactly the same kind of structure as common variants, we didn't want to just use 5 times 10 to the -8th. We instead kind of simulated it and tried to get what we think is a good threshold for a single phenotype, which came out to about 8 times 10 to the -9th for single variants, so pretty close to 1e-8 or so. And for genes, it ended up being something around 10 to the -7, 10 to the -8. So I'll talk about the full set of 3,700 phenotypes in just a moment, but wanted to dig into one phenotype to make sure that we were doing things right. So we ran an association test between one very readily available quantitative phenotype, height, and the genetic data, that is every individual SNPs, as well as SNPs aggregated into genes. These have been previously assayed by the GIANT consortium, and we replicate many of the effects that we find. So about for rare variants, for common variants, we were actually for both common and rare variants, we get about half of the variants replicating at genome-wide significance, and the vast majority, all but about 3 or so, replicating at nominal significance. So really a decent replication of this data set. Our data set is a little bit smaller than GIANT, so it's also not necessarily surprising that we may not have replicated all of them. The effect sizes are correlated with a little bit of attenuation in our data sets, as would be expected due to Winner's curse. They were discovered first in GIANT, and then we were replicating them, so you do expect a little bit of attenuation here. For the gene-based tests, we were able to use some of the results from GIANT as well, and we replicate about 9 out of 10 of their effects as well. So also pretty good concordance as we can find here. As with many big data sets, when filtering to the most significant results, if we were just to naively look at the most significant results in the genome, you'd often find lots of spurious results due to systematic errors or bugs, something along the way. This happens every time you look in an extremely large data set like this. So we devised several quality control criteria on variants, genes, phenotypes, and summary statistics to filter our data set to the highest quality set of summary statistics, which included about 40,000 group tests, so including LOF, missense, and synonymous tests, as well as about a set of about 1,300 independent high-quality phenotypes. So we also considered the idea that some of these phenotypes are not necessarily independent. So when we filter independence, we get quite a bit fewer. We do release the entire data set to the public before all of our QC, but we do provide all the QC flags as well so that anyone can do similar analyses as we do. So we can visualize these gene-based results in a sort of Manhattan plot. What I'm showing here should look somewhat familiar as a Manhattan plot, but instead of each point being a variant here, each point is a gene. So you might not see this kind of typical peaks of Manhattan plots as you might be used to. In this case, you do here in the HLA region, but some of these, just because there's one dot on its own doesn't mean this is necessarily a spurious result. When we're grouping into genes, we do see less of an LD structure because these genes do tend to be farther apart. So here for hypothyroidism, we find one set of genes significantly associated, the HLA cluster, which makes sense in the context of autoimmunity mediated at this locus, but several other genes that are kind of starting to pop up. But we didn't just do one phenotype, we did 3,000. So expanding from hypothyroidism, we could, for instance, do something where we collapse all diseases in the E chapter of ICD codes for all endocrine or metabolic diseases. And so now you see, again, this HLA is popping up, but there's several other kind of interesting loci potentially popping up for endocrine diseases as a whole. And so we didn't just do endocrine diseases as well. We did every ICD code that we could get our hands on, and we repeated the same for each ICD chapter. And now you can see, again, each of these is now a Manhattan plot kind of encoded in here, looking across all ICD code chapters. The HLA peak, you can kind of see jumping out on for many classes of diseases, which is obviously an important locus for human disease. But there's plenty more significant hits that are kind of popping up across the genome that sometimes is not shared across phenotype categories. I certainly don't have time today to talk about all the results for all 3,700 phenotypes. Really, I don't have time to do that anytime. So in our typical fashion, we released the full data set to the public this past summer, including all 600 gigabytes of single variant statistics and about 10 gigabytes of gene-based statistics. And alongside just the data dump itself, Matt Solomonson, who was also instrumental in working on the Nomad browser, has built an amazing browser to serve the data in an intuitive way with Manhattan plots and QQ plots for each trait, as well as also really cool visualizations of each variant within a gene. So you can search for your favorite gene, your favorite variant, your favorite phenotype, and explore the results here at genevast.org. So yeah, I highly encourage anyone to explore the data, download the data as needed, and feel free to do whatever you want with it. It's really a tremendous resource of associations. one thing that we kind of decided to do is explore a little bit of what kind of scientific insights we might find from this data sets, especially we haven't really explored, there has not really been a systematic survey of rare variant associations across the exome. So one thing we want to do is compare the frequency of loss of function variants that have significant associations with at least one trait. So, this is just loss of function variants. And the idea here is that as you increase frequency, you tend to have more variants associated with at least one trait, which is not particularly surprising due to power. Common variant, or as you get more and more common, you have higher power for discovery. So you do expect a kind of positive trend here. However, within each frequency category, we do find that loss of function variants have the most associations, followed by missense variants, followed by synonymous variants. So this follows kind of the expected patterns of deleteriousness for variation for individual variants. So you see this kind of same trend where they increase with frequency, but within each category, they increase with deleteriousness. We can look at particular genes as well and see that certain classes of genes are enriched for associations. So for instance, we have a set of known developmental delay genes, and we can see here that there are significantly more associations with loss of function variants compared to a frequency matched background set of genes. So these genes that are implicated in developmental delay are more likely to be associated with a phenotype than the average gene when we match on frequency. Again, as I mentioned before, frequency is an important feature here, so we have to kind of match this to a background set. When we look across gene sets, we see this pattern popping up for several gene sets, including genes undergoing natural selection. So here in these constrained genes, we see the same pattern. Known disease genes from a kind of curated literature of autosomal dominant and recessive diseases tend to have more associations, again, unsurprisingly, for both loss of function and missense variants, as well as several other categories like FDA-approved drug targets, genes in the GWAS catalog. This might feel a little circular, but it's kind of nice to see this this kind of validation set, as well as essential genes marked by CRISPR screens to be essential. Non-essential genes do not show any significant association. So finally, we can use this data set to explore several aspects of rare variant pleiotropy. We're still kind of in early days with this, but Wenhan Liu, who's an associate computational scientist in our unit, in collaboration with Luke O'Connor, has been spearheading the creation of a statistical test to detect pleiotropy among rare variants in the exome. So we're still, like I said, finalizing this test, but here's some preliminary analyses highlighting some putative independent effects. So I should say this is a little bit of pleiotropy and a little bit of thinking about heterogeneity, how if you have associations of more than one disease, the question here we're trying to answer is, are these associations mediated by the same pathway, maybe through the same kind of direction, or is there any possibility for heterogeneity, which suggests independence of effects? So for instance, here, when we look at loss of function variants, we do see a correlation, and so this is in this case, the ALB gene, which it codes for the albumin protein. You do see that loss of function variants are associated with decrease in albumin levels. That should not be too surprising. And they're also correlated with associated with decrease in calcium levels. This is a well-known kind of association here where these are kind of where decreases in albumin do lead to decreases in calcium. And so for loss of function variants, this makes a lot of sense that the effect sizes should be correlated. However, for missense variants, we see a much wider distribution, including potentially opposing effects as well, such as this variant up here that decreases albumin level, but increases calcium levels. So this potentially points to some kind of heterogeneity going on at this variant, where there may be some different effect happening for this particular missense variant. So with that, I'll conclude just by saying that, again, these data are now publicly available, this large resource for rare variant associations at genebas.org. I've kind of showed a little bit of a vignette about how the patterns of variant deleteriousness, natural selection, and frequency affect power and genetic discovery generally. And we're starting to use this data to identify pleiotropy among rare variants in the exome. That's not in the preprint, but it is something we're kind of actively working on now, finding these kind of heterogeneous variant factors that might point to detailed mechanisms. So with that, I'd like to thank everyone involved in the work, especially Ben Neal and Wenhan Liu, and Grace Tiao and her team for all the QC work, as well as our pharma partners, the HAIL team and our pharma partners for all their support. Of course, the UK Biobank and their participants, and the Broad Data Sciences platform as well. So thanks. Happy to answer any questions.
Thank you for that great talk. Yes, are there any questions? So we have a couple people raising their hands and in chat. So let's see, we'll start with maybe Pankaj. I'll unmute you.
Yeah, hi. That was a wonderful talk. Thank you, Konrad. And I'm curious about like what correction did you use for p-value threshold calculations for scatter? Did you use
Yeah,
bound for or Tukey or any other method? Or
yeah. So we
just curious
actually,
about.
so we didn't use Bonferroni. We weren't exactly sure what the correlation structure of the structure of the exome would be. So we instead kind of did it empirically. So I kind of glossed over it, but what we did here was we actually simulated a few phenotypes. So I basically just took random noise and simulated a few phenotypes, including heritable ones. So kind of heritable noise. We know the relatedness between individuals in UK Biobank, and we kind of spike in a little bit of heritability on our random phenotypes. And so when we looked at that, we can get a kind of empirical potential p-value threshold. So effectively, like create a QQ plot out of this random one, and the topmost point is kind of your divided by 20 to get at 0.05 is kind of a good potential p-value threshold for our empirical set. So we did that for the single variance as well as scatter and burden, and got somewhere in the 10 to the -8 range or so for the exome.
Thank you. Yeah.
Thanks. We also have a question from One Chain.
Oh, hi. So I have like two questions. Some of them might be like quite naive. So the first is like for those rare variant association tests, for those to be kind of like robust, how low the sample number could go? And also like the second question is how, what would be kind of like your suggestion in terms of like expanding this kind of analysis to whole genome data, especially in terms of like how could we best utilize the variant at like intergenic, intronic, or like UTR regions?
Yeah. So I guess to the first question, which actually let me answer the second question first because it's a little fresher in my mind. For whole genomes, we definitely it's definitely something I've been thinking about recently because I know we've been doing a lot of this whole exome stuff, and I know that a lot of genomes are coming online now. I mean, I think it's kind of hard because I think the most impact that we're going to get is still within the exome. Now, genomes give you better exomes as well, so it's not a bad thing to do a genome. It does seem like a little bit of a I think our money's better spent on exomes right now, but I know genomes are happening anyway, so we will have to kind of play around with them. So I think two things are going to happen. One is we're still going to do a lot of the same things that I just described on genomes as well. The non-coding regions are interesting because the average non-coding variant is less impactful than the average synonymous variant that I've been describing in this data set. And that's kind of our barometer for like not functional. So the average variant now is like even lower than that. So I don't really see a lot of burden tests happening that are going to be very effective to start, at least the naive, like the naive thing, which is the first thing that I would do too, which is look within every kilobase and start seeing, are there any associations with this kilobase? You might get lucky and get some common variants there, but then you're kind of just doing a GWAS again. There's another thing that we have been working on a little bit, which is I kind of mentioned constrained genes a little bit. I didn't talk about the whole concept, but in Nomad we've been looking a lot at regions of the genome that are depleted of variation, so less variation than you expect by chance. We term these constrained regions or constrained genes. We've traditionally mostly done this for coding variants, looking at loss of function variants of this metric called PLI, which I'm sure many of you are familiar with. We've recently expanded this framework to looking at non-coding variants, and we have a preprint coming soon, I would say in the next few months, that we're going to put out to explore what we can do about non-coding, about constraint, natural selection in non-coding regions. And I think that might start getting us towards the question of what regions of the genome should we be looking at? Are there particular, signals that might be a little bit more interesting? And And then, so I think that's one angle. And then the other angle is, generally speaking, we just need better methods for variant interpretation in non-coding genomes. We're still just not very good at it. You know, we can kind of talk about enhancers and maybe even motifs, but at the end of the day, you have too much variation or not enough variation, and it's still just a hard question, I think.
Yeah, thank you.
Sorry, you had a first question, which I forgot.
Yeah, I was just wondering for instance, for the rare variant association test to be robust, what kind of like the lowest sample number one can go.
Right.
Yeah?
I do have a plot about that a little bit in the supplement of the paper. I don't have it in the slides here, unfortunately. But the question is the question is, how rare is the variant and how rare is the disease? That's kind of the multiplication of those two gets you at how low can you go in terms of your sample number. With 500,000 individuals, a disease that's at 1 in 1,000 frequency, or 1 in 100 frequency, let's say. So you're now at 5,000 individuals, 5,000 cases, 500,000 minus 5,000 controls, 495,000 controls. So if you only have 5,000 cases, a variant at 0.1%, you only expect it in 5 cases. And even if you see it in 0 cases, you're not really gonna be, super convinced by that. Maybe if you see it in, in 10 cases, maybe if there's a, huge enrichment, then you might start seeing that. But that's kind of the key metric there is the expected number of alleles in cases. And if that's below 20 or 30, you're gonna kind of have a bad time. Now, because of that, you can aggregate variants across multiple, especially for LOFs, when you know that they're doing the same thing, you can aggregate variants in the same gene and try to get to maybe 20 or 30. But you're still, kind of at that, at that point. I think anything that's at about 10%, something that's at about 10% prevalence right now, we can get down to maybe variants or genes that are at about 1 in 1,000 cumulative allele frequency. But anything rarer than that, we're still kind of in a very low-powered regime, unfortunately.
I see. Cool. Yeah. Thank you so much.
Yeah.
If we also had a question from Biju in chat. He's asking, what is the biggest QC challenge when working with such a large data set?
I think generally our biggest challenge is managing the volume of data. When we're talking about 20 terabytes of data, traversing that data set, needing to touch every single genotype in a data set like that is very expensive, and just takes. We've gotten, thankfully, we have a longstanding collaboration with the Hail team here at the Broad, and they've been really responsive, and they give us great computational frameworks, and we try to break those frameworks with the biggest data sets possible. And that actually is a really good, we kind of nicely show them where the field is going and have with these large data sets. But there's always scaling challenges. I mean, even when we're talking about traversing these large data sets, it just gets really expensive. And finding tips and tricks to try to avoid having to do things multiple times is kind of really a big one. I don't think there's any one step of the pipeline that we're, that is more of a challenge than another. It's just a matter of managing all this and then trying to make sure that our time is spent thinking about scientific questions and QC rather than the waiting for jobs to finish, because that's really, and that really the Hail team is a big component of that. One way that we've kind of started to address this is it was convenient enough in previous eras of these data sets like EXAC and Nomad and even the UK Biobank, where we could kind of do one set of QC for the data set and move on. But now when our data sets are going to be in the millions, it is not feasible to redo QC every time you have a new data set. So we're developing the idea of this N plus K QC. So you have a data set of size N, and now you have a new data set, maybe smaller, maybe it's 1/10 of size of N, but let's say you for instance, with UK Biobank, we have 500,000, and now let's say we want to add another 100,000 on top of it. It's not really advantageous to re-QC the first 500,000 because we've already done that QC. So building frameworks where we can do K more samples, where K is smaller than N, and be able to do, and to do it efficiently and tack that data on in a seamless way. So we can always do it, but the question is, can we do it in a way where the data is still of high quality, where we're still doing a sort of joint calling process in order to learn the data, still seep information back from the case samples into the end samples, is still a bit of a challenge. And but we're building frameworks to do that. And I'm hoping we'll have something kind of at least conceptually done sometime this year or next year.
Great, thanks for that. That's interesting. I think Klaus has a question too. What's that?
Thank you, Konrad. It was a great presentation. I just want to follow I think this is a very interesting time because whole genome sequencing is coming. So everyone is wondering what is going to happen in association with, and how is going to do that. So I just want to dig a little more about this issue and say, question to you is, do you think the association was going to change a lot with changing the platform? First question, and then we.
I don't think so. I think these kind of frameworks that we're building.
It's a hard question. I know.
Yeah. These kind of frameworks that we're building like this, I think will look much more like what happens when we have full genomes everywhere. Well, so for instance, SageGene, this pipeline that we're running, actually does the single variant testing while it's doing the rare variant testing. So it can kind of do both at the same time. And we're even getting better at it now. And there'll be new versions of SageGene sometime, if not already, then very soon, that will allow you to kind of do arbitrary groups of variants too, which I think is nice. It already can do that, but now if you want to use, so we did in this iteration loss of function and missense variants, but now if you wanted to do them together, previously you would have to redo the entire association, which is not ideal. So now the new versions will allow you to do a little bit of kind of these combining methods, which I think will get really interesting and powerful for non-coding regions, where you can actually start doing non-coding tests without having to rerun these association tests. Because like I said, these are very expensive. The ones that we're doing here are about 4,000 CPU hours, or about $80 per trade. Not a big deal to do one or a few of them. If you have to, even if you have to do it 10 times or so, it's not that big a deal. But if you have to do it 100 times because you have different combinations of variants that you want, that's going to get really expensive for, and for not really much gain, because you're just kind of playing around with things. So having a method that does a little bit more combining of p-values and more combining of different variants kind of efficiently, I think will help us learn more about non-coding regions. But I don't see the whole framework really being torn up. I consider this an extension of the framework. I don't think this is that different from what we've been doing.
No. I was just wondering even the results. But for gene association, it is very important, the filtering. So what are you going to do for filtering non-coding SNPs? So, we have these splice scores, software, but we have to dig more to really separate these. I don't know what to recommend us, because we are wondering that.
Yeah, I don't think there's a good answer to this question. I don't think there is there's no best practice for this quite yet. You can use your favorite a composite score like CAD, for instance, and that'll get you some of the way. But I do think that some of these kind of so I think two things. One, better methods like that will of course be will probably win out at some point in the end. And like CAD or a version of CAD, whether it's CAD or a different algorithm, will kind of help us do a lot of that work. The other thing, as I just mentioned, these kind of like different methods of combining variants will allow us to kind of start exploring that a little bit too, and we can start saying, okay, if I could have a non-coding region and very quickly assess whether CAD is better or Linsight or some of these non-coding or like Arvis or something, some of these kind of variants that are these kind of methods that are looking for variant deleteriousness in the non-coding region.
Or in encode.
Yeah, exactly. And then if I can do 100 association tests in a few minutes just by combining different sets of variants and see which one does better for a single phenotype like height. We know what the answer should be in some cases. We have really good, we can assess things like heritability. We can assess things like association, replication with previous data sets. So we can kind of use some of the big phenotypes that maybe we're not trying to overfit. Like we'll use some of the variants.
Calibrate to that to go to that. No.
Yeah. Like so something like height I think is a good example because it's a good phenotype, but we don't actually then use it for any analysis down the line. So it's kind of nice to be a little barometer for what we end up using for for disease studies that we care about.
All right. Only this is a very silly question, but I think all the users would like to hear. What is the best association study software for you? So what is behind your pipeline? Because your pipeline is multi, but you use SCAT.
Yeah. So so we use so the pipeline that we use. It's actually, this is a great slide to be on. This is probably the best the best slide I could have landed on. We use Sage Gene. So it's really Sage under the hood is a generalized linear mixed model pipeline that uses a saddle point approximation to speed things up, especially for rare variants. So it is kind of a nice, so Sage is a really nice tool for this. It's very efficient, very fast, does a lot of the kind of things we need to, uses the genetic relatedness matrix. So it uses the idea that we have relatedness between samples in the mixed model so that we can use related samples, increase our sample size a little bit. We're not a little bit less concerned about population stratification. Still are, we're not throwing all of the samples together in one model. We're still doing separate models for each population, but we don't care that it's, we used to be very much focused on in the first iteration of the GWAS, we only looked at the white British subset because we wanted to control for population stratification as much as possible. Now we expanded to like the European subset. So it is still a single population, but not a very small sliver of that population. Like it is the whole population. So I think that's helped us out a lot. And the Sage Gene pipeline allows us to do the grouping of variants into genes or into annotation categories, LOF, missense, anonymous. So Sage Gene implements both burden and scat testing, and scat and scato. To be very honest with you, at the current sample sizes where we're at, I don't really find too big of a compelling reason to be running scat. I think the burden testing is getting us 99% of the way there with, and if we were able, if knowing what I know, I would have probably run just burden tests. Although at the time there was no Sage Gene implementation that only runs burden tests, but that is coming sometime this year. It may already be out. I'm not sure. It was a feature request from a while ago, and I think the author of Sage has finally has put it together. And that'll be great to use because that'll actually be way more efficient. My intuition is that the price difference between running just burden and burden plus scat is probably about 5 to 10x. So if we can save about 10-fold costs running just burden tests instead and get 99% of the way there, and then maybe you run scat or scato only on those variants that are nominally significant by burden, so that you can kind of do 2 rounds of it if you need to. Yeah. There are more modern methods actually coming out too that look at maybe something like deviation from mutation rate expectation, kind of like what we do when we look at constrained genes, but instead for phenotypes. I've been excited to try that method. I can't remember what it's called off the top of my head, but yeah, there's some new methods that I think will be exciting. Yeah, others have used Regini with some success as well, which is another similar generalized linear mixed model method. It has some speed ups that Sage doesn't, but it also has some, Sage has other speed ups that it doesn't. So I think, we'll get even faster with this as time goes on, for sure.
Of course. Thank you.
Great. Yeah, thanks. Great questions. If anybody else has any, please raise or put in the chat. I actually had one. This is kind of a hypothetical, kind of bigger picture question, but kind of along those lines. So let's ignore the cost of sequencing, storage, and analysis of data. And also sort of assume proportional inclusion of the different ancestries we have, to really get down to get most of this rare variant, rare trait associations, would you sequence, say just for the exome, maybe like everyone on the planet, 8 billion people, or do you think there's a lower number that would get us almost all the way there? Yeah.
If we're ignoring the costs, I don't see why we wouldn't just do everyone. There's no, if it's free, we'd obviously do everyone. I mean, I think it depends on the disease. I think we're already seeing GWASs. I think there was a recent giant GWAS just published for height that was like something like 2 or 3 million individuals, and that's starting to kind of really saturate the common variant heritability of that trait. So that already tells you that we can get good at common variants at 2 to 3 million individuals. We can get, sorry, not even good. We can effectively, I don't want to say solve, but we can really almost solve common variants with some sample sizes in the low millions, which is not unreasonable for most traits and diseases at this point. In the next 5-10 years, we will regularly see GWASs north of 1 million. That shouldn't be a problem for common diseases. So then the question is, how rare is the disease? How common is the disease, and how common is the variant? So if you want another, we're getting those, I think are getting to things like 0.1% frequency is probably pretty good for imputation for GWAS arrays, maybe 0.01%, so 1 in 10,000. If you want variants that are lower frequency than that, then you have to keep going up another order of magnitude to get all the way down. I don't think we really have a strong sense of the contribution of rare variants towards common disease heritability yet. We and others, actually, Luke O'Connor, a collaborator of mine, is working on this, and I think we might have some results sometime, in the next year or two about that kind of thing to really point out how important are rare variants towards common diseases, and then that'll give us a sense of what we need. I think it's going to be, if we can't, since we need 1 million, we need 1 million samples to look at common variants. I wouldn't be surprised if we need 10 to 100 million samples to look at rare variants. And then at that point, if you also wanted to look at rare variants for a, borderline not a rare disease, but something that's 1 in 1,000 frequency, then you kind of need 1 billion samples, and then we're talking about the size of the world. So it's, yeah.
So yes, we do need to sequence everybody, especially for rare diseases.
I think there will be strides along the way as we go. I don't think we need to get to every single person in the world, and we never will. That's just not reasonable too in terms of the infrastructure of that. But but I think getting a few more orders of magnitude will help us out a lot for sure.
Great. Thanks. I think Pankaj had another question.
Yeah, so I have one more question about the missense variants. Are you selecting for certain frequency in common population, or you're including every missense variant in that?
So we in this iteration of the data set, the SAGE gene actually implements we used every variant, and not just common or rare. We used all variants, but SAGE gene downweights common variants because we figure we've already first of all, we've already discovered them in GWAS, so there's no point in rerunning them. And second of all, they tend to be less deleterious, the common variants. The rare variants tend to be much more deleterious. So we downweight them by a beta distribution so that you really upweight the rare variants. In the next iteration, in the 450K, the final release of this, we actually removed the common variants. Anything above, I think, 5% frequency, we strictly removed, and then still downweighted everything up until there. So that we kind of really focus on the rare variants. We did play around with that a little bit. It's unclear whether that made a huge difference. I'm still kind of assessing that as we speak.
But yeah, I'm curious about the SCAT because I don't know how that affects SCAT analysis as well.
I don't think we've assessed it I don't think we've really done a full take of that yet.
Yeah, because I was exploring the GeneBAS data set, and it seems like there's a lot of information, but a lot of it may not be that useful. And I was wondering if we can sort of constrain those variants to certain filtering, may help a little bit more.
Definitely, and like I said, so I think for this iteration where we where we have these summary statistics, I think this is going to be kind of the best we get. But as these methods develop so that we can combine variants in arbitrary ways, we might be able to get even more information and kind of get some cool results out of that. But that's still to come.
Yeah, thank you.